Okay, so I wanted to start by looking at some of these project submissions today to make sure that we have some kind of an outcome from these ideas that you've developed over the course of this program, so that we can potentially, have it close enough to the something that you can actually do and finish in the next few weeks, hopefully over the summer, and have an outcome for this program. One second, something happened here with a drawing. Clear. Okay, so the first one I want to talk about is Trinika, your project, right, from lupus. Now, you submitted it some time ago, and we talked about it briefly last time. Did you have any updates on this project idea? Did you look through any data sets, or have you done literature review for your data set?
Well, that's what I've been doing lately. I really was trying to get the information that I needed to make sure. I was trying to get all the information that I need to put into my presentation and in my PowerPoint as well. So I've really been looking. I'm trying to stay on top of it to get all the information, the data information that I need to start my project.
Okay, so did you find a specific data set or not?
No, sir, not really. I pulled some where I reached out to the Lupus Foundation, and I have someone that I'm waiting on now to give me information from their trials that they've been doing lately. So,
Okay.
so I can see how to input that information into my presentation as well.
Okay, well, we did talk about this a little bit last time, so I kind of looked for some data sets. Maybe they're not what you want to do, but you mentioned lupus, so that's really the only thing that was guiding my my review. Here's a data set on Bioproject, right, where they were trying to explore the underlying molecular and cellular mechanisms of increased efficacy of the combination therapy regimen. And they had a mouse model of lupus and treated them with monotherapies of prednisone, mycophenolate mofetil, or tacrolimus, or with their combination, right? So they had here 1, 2, 3, 4, right? So 4 different treatments or combinations of these treatments. Consistent with previous clinical findings, combination therapy markedly improved renal outcome compared to the monotherapies in mice with lupus. Transcriptomic analysis of their kidneys revealed distinct molecular pathways that were differentially regulated in combination therapy versus monotherapies. Okay, so again, I don't know if this is exactly something that you were trying to, the direction that you were trying to go to. What's interesting about this data set is that they have a good number of samples, right? So 18 samples. They represent a number of different conditions. They've discovered some differentially expressed pathways, right? So a lot of these things actually, sounded to me like they might be relevant to what we discussed in this course. I just want to make sure that, it's clear. Let's say if I was submitting this project, what would that look like? And maybe as we go through the session, you can make some notes and, potentially maybe like by the end of this week, by Friday, tomorrow, or maybe Sunday, you can, rephrase some of the wording in your project. So let's go to the form and just kind of see the different elements and how to fill them out. So the first one is problem statement, right? Then we have project summary, link to the raw data, and reference publications. So the problem statement, right? Let's think about what is the problem statement that they were trying to explore here. What is the, what are the specifics that they were trying to find out? Trinika, what do you think?
I'm just looking at it, rereading the information that you just spoke upon.
Okay, does anyone else want to summarize what the problem statement here is? Okay, so, to clarify, they give that problem statement right here, right? So they were actually looking at specific mechanisms that were useful, or they contributed to making this combination therapy more efficacious or more efficient for lupus, for treating lupus, right? So they probably have some clinical outcomes, and they can say, okay, the combination worked better than the individual therapies. They probably have a control there, right? But that's basically their idea. Their idea is to try to understand at the molecular level how combination therapy is working differently from individual therapies. Now, let's say I have this data set. I don't want to replicate exactly what they did, right? So I might want to be more specific as to what I would want to focus on. So I would go in and I would say, here are these 4 different drugs, right? We have here, 1, 2, 3, sorry, 1, 2, 3. Okay, so yeah, 3 drugs or their combination. So what I would probably do is I would research a little bit more to understand what these different drugs are, right? It's not very difficult. You can probably just do a search on it, right? So where are they? Okay, so let's take this one, right? And just to understand, right? So at the molecular level, what exactly is the type of this, treatment that we are talking about? So let's just look this up. Okay, so these are immunosuppressant drugs, right? So they actually reduce the strength of the body's immune system, right? And they are treatment of several autoimmune diseases. And now it becomes a lot more interesting, right? So if you just look this up, you start understanding that, okay, so we have this immune suppression, right? And the immune suppression potentially could be combined with some of these other types, right? So let's see what this one is. Okay, again, we have different immune system disorders, right? And this is a corticosteroid. So again, we have 2, but they might have different mechanisms of action, right? So the question would be why a combination of these 2 would work better than them by themselves. Let's try to see what this last one is. Okay, also is an immunosuppressive drug. So we have 3 different immunosuppressive drugs, but this one, you can see that it probably works on different types of cells, T-cell mediated diseases such as eczema and psoriasis, right? This one, typically when you look at them, they will tell you a little bit more what the, what does it actually do, right? So let's look over here. Okay, so this is kind of that process of doing, the context. Let's set the context, because ultimately we also want to look at the specific data sets that we have to try and see what type of data could actually be answering this question that we might have. Okay, so side effects. Okay, so here, I think it's interesting with the immune system, right? Because the immune system is good for you. And of course, you can have different kinds of disorders, but you can see here that it mimics the effects of hormones your body produces naturally in your adrenal glands, which are small glands that sit on top of your kidneys. So you can regulate these glands in your kidneys, and they will produce corticosteroids that suppress inflammation, right? And they change the way that this tissue works, right? So now let's take a look at the data. So the data here, you can see, has 18 experiments or 18 samples. So let's take a look at what are these 18 samples. We can either look here at the SRA links, right? And here you can see different replicates like 1, 2, and 3. H probably stands for healthy. And then you have 2, right? And different drugs right here. So you have different samples, right? Those different samples are treated by different mice that are treated with different drugs. And now we can design our analysis, right? So one straightforward analysis, if you want to replicate exactly what they did, you just want to say, let me take different comparisons from the different drugs or combination of drugs, like multi probably means combination, and then see what are the different genes that are differentially expressed and how are those genes organized in their pathways, right? But sometimes you might want to say, okay, but this is their project. I don't want to replicate exactly what they did. Could I do something a little bit different so that I can actually focus on, something that they did not discuss? How can you do that? For example, if you look at this project, they don't have a publication, right? But if you keep on reading here, combination therapy inhibited TLR7 expression in the kidneys of mice. Combination of tacrolimus, led to better stabilization of the podocyte actin cytoskeleton through the reciprocal regulation of RHOA and RAC1 activities. Combination therapy strongly suppresses the IL-6 STAT3 pathway, right? So this is that, cytokine immune pathway. So these findings were further validated in renal biopsy samples from patients with lupus nephritis before and after treatments with these drugs. Thus, our study further supports the earlier clinical finding and further provides insights into molecular basis for increased efficacy of combination therapy, right? So we know that we can look for other data sets that might have lupus, and let's say, what's the role of this pathway in lupus, right? Okay, and look, you have, okay, that's the one that we looked at. Let's look at other publications. Okay, so now you can see how the specific pathway actually contributes to dependent Th17 dependent autoimmune diseases via epigenetic regulation. Let's see, effects of histamine on antioxidant activities and regulatory T cells in lupus-prone mice. Okay, that looks interesting. So you can actually start looking at how same pathways and same treatments were treated, because you might want to understand, right? So when we talk about immunosuppressant drugs, typically the result is, this person has some manifestation of the disease as a result of this autoimmune response. And when we treat that response, it's actually just taking away the manifestation of the disease. It doesn't actually change the underlying issue, doesn't address the underlying issue. So you might want to compare how, let's say, early stage lupus, late stage lupus, there are different stages to the disease, there are different severities of the disease. You can start looking into how this pathway changes in different conditions between different types of patients. And you might want to think, well, okay, can I look at these pathways or the specific genes in different cells and develop some kind of a sense of how molecular information can guide the selection of treatment, whether it is one type of drug, one type of a combination, another type of a combination, right? And how does it actually affect the different types of manifestations that this disease might have in different patients? So if I was to focus on lupus and develop an independent project like what you're trying to do, I would actually not try to replicate exactly what they did, but actually take some of the data sets, research this topic a little bit better, and maybe combine a couple of different experiments that I can then talk about in my analysis. So that's, in terms of developing that research question, right? A little bit of literature review and looking at what literature says about these findings or something relevant from the disease manifestation can give you great ideas about how to combine different data sets and develop something new. The other part of it, right? So once you do that, right? So now we've developed some kind of a problem statement. When you talk about your project summary, right? One of the immediate things that you'll find is that if you want to not do a literature review, a review paper, you want to do a research project. In the research project, you have to have data that you understand how it was generated. So in this first example, we saw the SRA files, right? And these SRA files describe how these different, samples were generated. So what do we know about how they were generated? We know that they were generated, using specific mice. You can see here the type of mice that they used. And you can see here the different treatments that they got, right? So here you have MMF, right? And so you can organize them into groups and start comparing them. And there are two ways that you can do that. One way is by reproducing the whole analysis that they did by doing, for example, downloading SRA links and loading them onto the platform. Or you can actually just take the specific geo data set, right? Sorry, it's the wrong one. If you go back to the original page, okay, you'll see that you also have a geo data set. So what is a geo data set? A geo data set is data that has already been processed. And you can download this data in TXT format. So this TXT format will, here in the raw data, will look like this. Okay, here I have them downloaded, right? For each individual one, you see that you have the name of the gene and then its expression, right? So I can look at, here I have some of these expression for normal files, right? And then let's say I'm interested in, I don't know, prednisone, right? So I can create a table, and let me just quickly show you how to do that. Okay, so here you have, normal or prednisone. So this is 2. Let's take another one from prednisone. Okay, these two columns should actually match, right? So this is just the name of the gene. This is the name of the gene, this is the name of the gene. So if you scroll down, you can see that they're in the same order, right? 13, 14, 22, 13, 14, 22. Scroll down and take another place. So let's go to 400. And just to make sure that they're the same. Okay, so this is 23 Rick, right? So 400, 23 Rick. So it looks like they're the same. So you can just take this column, right, and paste it over here. So here now you have a one condition, prednisone, and yeah. So you have 2 samples from prednisone, 23 and 29. Now we can take some of the normal ones, the non-treated, right? So this one. Let's say this one. Again, we just create a table of expression without having to process all of the data. So here I have this one. And here, this is pretty soon. Now, 2 samples are not enough. Remember, we talked about that, how if we just have 2 samples in each group, it's not enough. Because what if this and this do not agree? They are mostly the same because they are replicates, but they might not agree. So if they don't agree, I need a tiebreaker. So I need a third one. So let's close this and get just another one. Okay, this is just one type of analysis that you can do. So we're just going to try and do this fairly quickly. Okay, and let's get another one for prednisone. Prednisone 35. Okay, and then I'll show you how to quickly run differential gene expression if you want to, try and look into differentially expressed genes. So, okay, so here you have, right, you save this as a GSM 53021. So GSM or GSC, right, 5153.021, and leave it as tab delimited file. Okay, this is a table of expression. So Trinika, really quickly, did you have a question on how to create this table, or was it pretty clear what I did?
It was pretty clear the way you was doing it.
Okay, so now let's go take a look on the server platform. You have here what's called differential expression. Differential expression really requires just this data set that we just created. Okay, this table. Once it's loaded, you got to give it just a couple of minutes for it to recognize the names of the samples in your top row, right? So then you click here, and here you have those top rows, right? So you have the healthy, or let's put the healthy right here, and you have the prednisone. So you want to compare this group of samples and this group of samples and find those genes that are differentially expressed. Okay? Here you have a few settings. Now, the important setting here is that you're using the mouse genome, not the human genome. So you can't leave this human. You're not going to get any results. Here you have some other settings. You can play around with those later on, but for now, I'm just going to skip that. Okay, and that's it. So really just one step that you can do. Okay, so we'll call this lupus. Treatment differential expression. Once you have the analysis results, and again, this type of an analysis is not going to take long whatsoever. But instead of waiting for it to completely finish, I'll quickly show you what the analysis results would look like in terms of what types of files are you actually going to see. Okay, so first of all, you will see all of your genes organized. So let's take a look at this file right here. You can see that all of your files are organized with a number of parameters that this pipeline puts out. And the parameters actually show you the p-value, right, and the p-adjusted value. P-adjusted value is p-value adjusted for false discovery rate. So do you know what this p-value shows, Trinika?
You say that again about the p-value?
Yeah, do you know what it shows? What does it mean that it's 0.09, for example?
No.
So the p-value is your probability, right? So how probable is it that you found a difference between those two groups, right? You were comparing one group versus the other group. So if it is probable, so by chance you could have found it, your p-value is going to be high. If it is improbable, so it's not likely that it would have happened by chance, there's a small probability that it happened by chance, that means that it really represents a biological difference. So you're looking here for small values. So, but false discovery rate accounts for, thousands of observations that you have. So instead use p-adjusted value, that will be more stringent. And now you can just sort them from lowest to highest. And your cutoff is about 0.05. That's just a rule of thumb. Basically, it means that you cannot be mistaken by more than 5% of your data. Okay, so somewhere here there's going to be 0.05, and all of the genes above are actually differentially expressed. Okay, and then you have some additional outputs. So I see that your pipeline finished. Let's take a look at what we got in your analysis. We didn't get anything, okay? So why do you think we didn't get anything there when we got something in this other one? The reason typically is because the data set that you provided actually has an issue, okay? So let's see what kinds of issues could be here. Okay, so if you look here, right, there might be a few different issues. One of them is that you have weird characters in these descriptors above, okay? Another one is if you look through your genes, you might have some weird characters here. For example, there might be a 0 somewhere, there might be a dot, there might be, something else. There might be a missing value somewhere. So those are, or here you can see there's a dash. So these are bad characters. And it's very important to not just kind of get to a dead end and really explore your data because of these technical issues, right? So a huge part of bioinformatics, always technical issues. You always have to look for something. Okay, so what I'm saying is, here's an overview of how something should work, right? In this analysis, everything is going to be very nice because, somebody took the time to review the data and look at, here you see a whole pathway, right? That is regulation of actin cytoskeleton with differentially expressed genes. So now all you need to do is you need to say, okay, this pathway is already enriched with differentially expressed genes. What does differentially expressed genes mean? It means that between the first group and the second group, there's a difference. What kind of difference? All of the green ones are down-regulated in the second group, and all of the red ones are upregulated in the green group. So if it is, right, so the other way around. So 1, it's upregulated in the first group, -1, it's upregulated in the second group. Okay, so then you can start looking at this and try to think, okay, biologically speaking, what could this mean that this actin cytoskeleton regulation pathway is differentially regulated when we apply this treatment? So this is a brief overview. We're not going to spend all of our time just talking about your thing, but I can actually probably help you figure out what are the issues with your data set, make sure that your data set works, and you can take it from there. So hopefully after this review, what I'm looking for really as an outcome of this is if you could modify some of your form submission to a level where what is it that you're looking for in this data set, what is the data set about, and once you find whatever you're looking for, it's going to give you some new information that you've discovered, and you can clearly identify where you got the data set and how you're going to analyze it, right? So for link for raw data, for example, you would put in this data set that we were looking at here, right? Which was that GSE collection of files, this one. So you can put this or the bio project even, you know, as your, so even this, right? This is something that you can put in as your raw link to the raw files. Okay, so again, the only thing is that we gotta make sure that, for example, this data set is, oh, you know what the reason is? The reason is that this is supposed to say ID and not symbol, and then just eliminate some of these weird characters. So really that's all the issue. And you know, this is a simple analysis that with a little bit of practice in a few days, you could probably complete. And if you have any questions or, you know, challenges with this, you can actually ask about those.
Yes, sir. Thank you.
All right, great. So the other project submission that we just got and we haven't had a time to review was Roland. So we'll get to you in a second. But Wineen, did you want to mention something? Mention something about your data set?
Yes. I went back and I went to the NCBI website and I typed in organochlorine pesticides, and what came up was the data set for comparing different pesticides in some was aquatic environments. And so they had this one comparing two pesticides together, and I thought which was pretty neat was para DDE, which is a metabolite of DDT, and methoxychlor. And they have two data sets that they actually look at. And so because the pesticides are considered to be endocrine disruptors, they interfere with, you know, hormonal activity. And so I thought this was a pretty neat study. The others I looked at, it really didn't give me that good comparison like this one did. So I was pretty much focused on this one. Hopefully that this may work. I don't know.
Well, yeah, no, interesting data set, because first of all, let's look at your description here, right? You originally, this was your original submission, right? You were looking at transcriptomics and cytokine production caused by pesticides and viruses, right? So when we were talking about this problem, we were looking at pro-inflammation cytokines, or in general, deregulation of inflammatory pathways, right? I think narrowing down to a specific type of a cytokine or something like that is going to be difficult, but we can actually look at deregulation of immune function in general. So if you go back, and by the way, when you submit, you know, your data, so like you did here, you just put in the terms, right? But instead, you know, it's a little bit difficult to know which data set you are referring to. So I would prefer if you can put in, you know, the geo, or you could look at the bio project, but a specific one, right? So that will give us all of this information to take a look and see what's in there. So, you know, I would put this right here. Okay. Now, if we look at immune response, right, let's just read this real quick. They were looking here at BAS, and, you know, they were trying to assess the impacts of these two pesticides. And then they were looking at males and females, right? And they have led to separate group of LMB, okay, after 84 days. significantly incorporated them right into muscle and ovary tissue. Gonadal histology and plasma sex steroids were not significantly different than controls after being fed contaminants. Microarray analysis revealed that this DDE and fluid regulated more genes in common than either chemical compared to MXC, consistent with data that is more potent anti-androgen than MXC. And the interesting thing for me is, at the end here, so cell processes affected by all three treatments included leukocyte cell adhesion, ossification, platelet function and inhibition, xenobiotic metabolism, fibrinolysis, and thermoregulation. Most striking was that all three chemicals depressed gene networks related to immune function and inflammation, right?
Mm-hmm.
So immune function and inflammation is obviously related to viruses as well. The biggest challenge here, right? What do you feel is the biggest challenge about, answering your question with this data set?
The species, would it be that it would be very different, and I'm going from now a fish to maybe a mouse or something like that?
Yeah, I agree. If you have a fish and you find some regulation in the fish, does that actually mean anything about the human? And the other way around, if you have human viruses, right, who knows how they actually work in fish, right? So if you wanted to answer this question, you would have to find a fish virus and then have a data set from fish that talks about that viral infection. So if you can find the same thing in mice or in rats, right? I saw a few from the previous link that you gave here. I saw something rat related.
Mm-hmm.
Let's see real quick.
They do, on the same page, they have a lot of mouse, where they talk about sprayed daily rats that they use in their experiments. So they have a lot of those experiments as well.
Mm-hmm.
So now I changed my focus from looking at the gene expression from, in that study, they were just using female largemouth bass. They were not using male. So and because the pesticides can cause deregulation of certain pathways. So I was just looking at, would it make sense to compare the two since both of them were organochlorine compounds?
You mean in mice or do you mean in fish?
In fish.
Well, the problem with the fish, there's another problem also related to their species. I don't know how good is the annotation for bass. So, I mean, maybe, you can try. So again, you kind of saw what we did for differential gene expression. So here, right, you have all of these samples. There's 16 samples. Here is your raw data, right? If you
but that file is so big.
Yeah, unfortunately it's big, but that's what big data is all about, is big files, right? That's a joke. So, this is, I think if you open it up and you kind of clean it up, you don't probably need all of the 16 samples. The problem is this though, if you look at this, it's going to have, two issues with it. One is this is microarray data, right? Let's take a look at it. It has probes instead of genes.
Oh, Lord.
Good thing, but look at the annotation. The annotation here is unknown, right? Description unknown. So you have here a bunch of genes that are unknown. Well, all of them are unknown, right? So the way that you would interpret it is you would have to find a fish expert and sit with them and really understand, you know, what is it that you know about this specific region, or, you know, here's one that is actually maybe known, but, you know, there's probably not much known about it. So in general, you know, these non-model species are an additional set of challenges that, you know, is difficult to overcome. So probably for this, it would be easier to work with mouse, human, rat genomes. That would
Okay.
probably be the best thing.
Okay. So what you're saying is what, doctor? So what, I need to go back to my original idea?
Well, I think, you know, if you take this as your research literature review, right, you found that some of these pesticides are related to immune function and inflammation. And for me, that's a confirmation of your original idea that says that, yes, this is worthwhile to explore. Now, how do you go about exploring it next? It looks like if you go back here in the search, right, and let's do the search again. Okay, it looks like specifically for array, yeah, it looks like the only one for organochlorine is this one. And this one is increased risk of Parkinson's disease. Yeah, this is changes in DNA methylation. So changes in DNA methylation would again require, you know, you would want to look at one of these and see what is, oh, okay, look, they actually provide you with an Excel spreadsheet, looks like. So just like it's difficult to define your research question, it's also difficult to find all the right kind of data. So here, yeah, they show you the different conditions in Excel, which is neat. So you can see here that, you know, they were looking at midbrain tissue from mouse, right, and looking at these different characteristics like age, sequencing method. So it looks like they did RNA-seq. You know, this is, oh yeah, you have a summary right here of what they found. But this is not necessarily gene expression, right? This is epigenetic regulation of gene expression. So I would suggest doing a little bit more searches. It looks like, you know, just like you came up with that search term for organochlorine and for pesticide, was it, right? You could probably think of some other pesticides or some other combinations of terms to look for.
Okay.
Yeah.
I will do it. Thank you so much.
Yeah, thank you. And let's just take a brief moment. Roland, are you still around here?
I'm here.
Awesome. How are you doing?
I'm good. How about you?
I'm doing great. So here's your project idea here. You've got comparison of A blood samples with Zika virus to protein sequences in Nigeria. So could you expand on what you think you want to do?
Repeat the question again, sorry.
Oh, if you could just, you know, expand on this, tell us what you have in mind.
Well, like upon looking on NCBI and typing in Zika virus, I was just gonna focus on like the African region because like I believe that's where it's the most prevalent. And upon searching the database, I found one like it said isolation and it said blood, so I clicked on that and it brought me to a link. If you scroll down, not right there, but scroll down. It's isolation site source. I'm sorry, isolation source.
Yeah.
And you see that
Publication.
yeah. Yeah, I was planning on using that one blood isolation source as a reference database to compare like the 7 sequences of Nigeria to it to see if like there's like what common mutations there are from frame shift, like like if there was a point mutation like a deletion, a substitution, or a frame shift mutation like an inversion.
Cool. Okay. And what's your question?
Like, how did these like? What effects does the Zika virus have in that African region? Like, because like normally Africa
So you
is
wanted to
like.
compare. So sorry. So here you have sequences. All of these sequences are from Africa. Yeah. So you were trying to compare what African sequences to non-African sequences.
I could do that, yes.
Because here, just a little quick issue with this data set, you can see that it is, these are incomplete sequences. Remember how we looked at on NCBI virus, you can go to nucleotide completeness, and all of these are actually partial, right? But if you look at sequence length, well, I guess you can't do it from here, but here you can see the lengths. 2 of them are comparable lengths, but these are all smaller regions. And if you actually do a line, which is what I did right here, see these sequences are here, this sequence is way over here, and these sequences are right here, right? So this data set by itself, if I was to use it for multiple sequence alignment and let's say compare African and non-African, you would actually want to probably add here, I don't know, Brazil, let's say, because they have a lot. Unfortunately, this filter here doesn't work that great. But here's some of your Brazil, right? And again, you've got the length. Just take a look at, what lengths you have. So if you look at the complete lengths, right? Yeah, so the protein sequences, let's say the complete ones, this one is about, no, I think, yeah, this is the complete one. So this, oh, you know what? Sorry, we were looking at protein sequences. Were you going to look at a specific protein, or were you looking at, because it looks like maybe these are different protein sequences. This is envelope protein. What is this? Envelope protein partial. Envelope protein. Polyprotein. Yeah, polyprotein. So these are different proteins. Were you going to focus on a specific protein or no?
Now that you mention it, like that would be better. Yes, I will look at a specific protein.
Okay. Okay, but I think that, the idea of comparison makes sense. Now, after you find some, common or different mutations between, let's say, Nigerian and non-Nigerian, what would that lead you to make a conclusion about? Like, what do you get out of that comparison?
Well, I could get like, because Africa is like heavily, because like basically like where that region is in terms of like, geographic location. Because I mean like, because I mean, if we're talking about Brazil and Africa, like I believe like they're both below the equator, so like the climate has something to do with it, as well as like Africa being more like I wouldn't say like it's more heavily woodsy, so like the mosquitoes could like more mosquitoes in like a heavily woodsy area could play effect in it, as well as like and like access to sunlight does play a role on genes. And I know mosquitoes thrive in sunlight, so or regions where there's like a lot of sun, yes, heavily sunlit areas. So like that's the conclusion I would come up with, like basically geographic region, climate, and access to sunlight.
Well, maybe then it would make sense to find some, not sunny location, right? So maybe, here instead of, I mean, maybe Brazil could be like, it has different something, but maybe it's not, that different. So maybe from Europe, it looks like you've got, let's say United Kingdom, definitely a different climate, right? So here you've got United Kingdom, unfortunately just one. Maybe we can just say Europe in general. So Italy, probably not as, probably different also. So you've got 4 here. These are complete sequences, and you can either look at specific proteins, or, you can, if you deselect that, depending on, like, there you go, Russia, that's probably very different. So you can probably prepare a better comparison by researching a little bit more into here. And it sounds like out of the many possible factors, have you done any literature review on influence of these environmental factors and Zika virus?
Oh, have I done any research?
Literature review.
Is that what
Have
you're
you
asking?
read any literature?
No, sir, not quite.
So that would probably be helpful also if you go on, actually about the Zika virus, there is a dedicated site all about Zika. So you can see, infection and molecular effects on host and data collection from PubMed articles. So there's a lot of stuff here that you can actually look at. It's, and there's a few things that you can, learn here from the different proteins, right? So you can search for different proteins and kind of see how they're related. And there's also, one sec. So there's a virus database, just to kind of see, I forget. Yeah, there you go. Viper. So here you can look for Zika virus and learn a little bit more about, there's Zika virus right there. Okay, they have a lot of information on kind of, the genome, the different proteins, what is their function, maybe even you can find, so there you go, you can see an interactive map. So it looks like, the majority of it happened here. See, it looks like it started here in Brazil, and then you have all of these infections that started spreading. And I bet you some of those African sequences that you found are much later. So anyways, you can see this. I suggest looking and learning a little bit more about the Zika virus in general, and just also looking at the different proteins if you're going to be selecting a specific protein. They also have data sets here. Right, so take a look, and if you have any questions, you can also let me know. Okay, Roland?
Yes, sir, I'm here. I'm just copying down this link.
I put it on chat for you, if you can see it on chat. Okay, so
Okay. Yes, sir.
so let me know how it goes. Yeah, did you have a question?
Yes, like, one quick question. Like, I'm kind of confused. Like, once I do complete the project, like, how will I do the project submission again? Like, how do I submit the project and who to?
Oh, yeah. So you actually go on the forum, right? So the idea here, by filling out this form, you are defining what your project idea is and coordinating how you plan to take what data, making sure that the analysis that you have in mind will work for the data that you found. After this, you will prepare several slides that will address each one of these parts of the project. So you'll explain a little bit, this is the data set that I found. I found 5 sequences from Brazil, 5 sequences from Russia, 5 sequences from Nigeria. I want to do multiple sequence alignment and maybe a phylogenetic tree, see whether they are different. Once I know that they are different, I am going to find specific regions where they are most different, right? So then once I find those specific regions, I will look what proteins are they in and try to understand what biological function might these differences have because of different mutations. And then the question is whether what you found, the number of sequences that you found, is enough to establish some association with other differences in patients or in the climate in those different countries. So, if you kind of start preparing a slide for each point, so this is my analysis, this is my data. In my data, I have 5 groups. These groups represent these conditions, or they represent these situations that I'm going to compare. And then you have, some literature review that shows that you understand, once you find those mutations, where they are and what their impact is and things like that. And at the end, you'll end up with a presentation. That presentation, we'll review it together, to make sure that it's ready to be presented to somebody else, that you have answered all the questions in a way that you know where you got what and what it means. And then we'll send it to an external reviewer after the program is over. So that external reviewer will have some expertise in this domain. They will comment on your presentation, suggest, where it was good, where it could be improved. And that will be the outcome of your program. You have completed an independent project. You've presented it to a peer who is also in the research field, and have incorporated, the feedback that you've gotten from them and refined your presentation. So then after that, you will record your presentation as a video and we'll upload it in your group so that you have that project completed.
Okay. Will Google Slides work?
Yeah, Google Slides, PowerPoint, Keynote, whatever works for you.
And then once I'm done in Google Slides, I just upload it to the project submission.
Well, you just copy a link, right? And actually just put it on the forum. So whenever we have this forum right here, right? What I'll do, we are done with sessions like teaching sessions, right? We are now working on projects. So here we will actually start project submissions. Okay. And here you can paste a link. Okay, so does anyone else have any questions about the project submission?
No, sir.
Okay, All right, so if you want to, submit your draft, go ahead and do that here. If you have any questions, you can also post it here. If you want to coordinate a time to speak about your project data, for example, you want to get help on your data or something like that. Yes, the courses are going to remain open for the next month or so. I forget the exact date, but there's an expiration date here somewhere. Let's see.
August 5th.
Yeah, so you have it until August 5th. August 7th, yeah. All right, well, it was great to see all of you again today. Hopefully, if you don't have a project that you're working on, you still have until Sunday to submit something and then start working on it. And after that, we will actually close that main forum. So, you won't be able to submit any new projects, but you can still work on, other projects and coursework. And right here, I believe we should have some projects as well. We are working with a number of teams that have developed a project on COVID-19, Ebola, and tuberculosis. So hopefully before the end of the month, you will see those appear in here, and I will let you know in the forum also if those become available before then. All right, well, thank you all for joining today. I'll see you again whenever you have an update from your project.
Thank you,
And
sir.
Pranika, I'm actually going to
Thank you.
I'm going to check this data set and email it to you after it works, just in case you want to use it.
Yes, sir. Thank you.
Thank you. All right. Take care, everyone.